DNA viruses generally rely on the host cell's RNA polymerase to describe the viral genes into mRNA. Once the viral mRNA is made, the host cell treats it kind of like its own messenger RNA, and it's going to translate it into viral proteins. So that's what we see here, right? The host genome is going to have double-stranded DNA, and so the viral genome can kind of just jump right in and use the same process and use the same kind of equipment, the same enzymes. All right, when we have single-stranded DNA, like some viruses do, there's an additional step to kind of get to the point of transcription or replication, and all that's going to happen there is that single strand, the host's DNA polymerase is going to build the complementary strand. So you have a double-stranded DNA, and then it's just going to act as double-stranded DNA for replication, transcription, and translation. Um, so those are DNA viruses. RNA viruses are going to be a little bit different, right? They're already RNA. Animal cells don't have enzymes that can copy RNA into RNA. So the RNA viruses must bring or uh, the enzymes with them, or many of them do, or at least have a gene to right away make a different polymerase. Um, and so for each of these different types, you can think about, what type of genome does the virus have? Which steps can the host cell perform? And which does the virus need to have kind of some special equipment to get accomplished? All right, and so if we start with the positive sense single-stranded RNA virus, um, this genome, it's already in the kind of correct 5 to 3 prime direction, um, which is the same as messenger RNA. So because of that, the viral genome, it's the same sense as the messenger RNA. As soon as it enters the host cell, translation can take place. So it can be used directly by the ribosomes to make the the proteins that are on those viral genes. Um, and in this case, one of the first proteins it's going to make is this viral RNA-dependent RNA polymerase, also known as replicase. And so it's an RNA polymerase, meaning it's going to build an RNA strand. It's RNA dependent, which means it's using RNA as its template. Um, Animal cells don't have this enzyme, but so any positive sense single-strand RNA viruses are going to need to have a replicase gene on them. Um, so with this one, for replicating their genome, it's just slightly more complicated. And so the virus is going to use that replicase now to make a complementary strand. So that'll be a negative single sense RNA molecule. Um, and then it can use this negative strand as a template to build um, the more of the genome, so that positive sense single strand RNA. And because it's building complementary RNA from an RNA template, it is going to use the replicase that it made to make more copies of that genome. Okay, for negative sense single strand RNA, um, it cannot be kind of directly translated. It can't just jump right in like the positive single strand can. So it needs to bring replicase with it. So it's part of kind of the virus. It's bringing replicase that when it was originally made, the replicase was made in that host cell, and now that it's infected, a cell is going to use this replicase. And so it'll make the complementary strand the positive or the sense strand, which is the same as messenger RNA, the mRNA. That'll be translated. It can make more replicase that it's going to package up and send with the new copies of viruses that it's going to make. Then so that's making proteins. When it's replicating its genome, again, it's going to need the replicase. And so it's going to use that complementary strand and again use replicase to make more of the genome that's going to get packaged up for the new viruses. And so the key idea here is that the negative sense RNA viruses need to bring replicase with them. The protein production and genome replication both are going to depend on that. And then a little bit about retroviruses. So these kind of break the usual flow of information, right? So instead of what we see in typical animal cells going from DNA to RNA, they have a enzyme called reverse transcriptase that is going to kind of go backwards. So it's going to take RNA as a template and it's going to build DNA from it. All right, so if we think about single-strand RNA retrovirus, this is positive sense. It's going to have reverse transcriptase with it, and that reverse transcriptase is going to be, rather than a DNA polymerase or a RNA polymerase, it's going to use reverse transcriptase to build a negative sense DNA strand off of the positive RNA strand. After it builds that, it will use the regular host DNA polymerase to make the second strand. Now you have double-stranded DNA. And from there, it will go through protein synthesis, and genome replication, similar to the double-stranded DNA viruses or what happens within the cell typically. Another kind of important thing with retroviruses is in this double-stranded DNA state, it can become integrated in the host genome. So it's not necessarily making copies of itself that are going to butt off of the cell. It could just remain latent and be copied as the cells go through their cell cycle and make copies of themselves. All right, there are retroviruses that kind of start off with DNA as well. I won't go into that too much, but they can replicate their proteins just using the host information. But when these ones do start replicating their own genome, they're going to go from the viral DNA and then be transcribed into RNA. Then they're going to use reverse transcriptase to go back to DNA. So it's not a particularly efficient pathway, but they end up relying on reverse transcriptase because of this kind of pathway that they use.